Welcome to the Nourished Child Podcast, a show about childhood nutrition, feeding kids, and dealing with the ups and downs of growing a healthy child. Here's your host, registered dietitian and childhood nutrition expert, Jill Castle.
Before we get this show started, I want to share a message from our sponsor. Drinking 3 glasses of milk a day is part of a healthy diet, especially for young children. With 13 vitamins and nutrients, it's hard to beat milk's nutritional value at any age. For more information, visit southwestdairyfarmers.com. A healthy gut and gut health is a hot topic these days, and today we're talking about this topic, specifically building a healthy infant gut, with my guest, Dr. Noel Mueller. Noel Mueller is an assistant professor of epidemiology at Johns Hopkins Bloomberg School of Public Health, and his interests and work is in the primary prevention of obesity, type 2 diabetes, and cardiovascular diseases from the perspectives of life course and nutritional epidemiology. His research has focused on the determinants of the mother-child exchange of bacteria and how this might influence health and disease risk. He's published numerous research studies, and I invited him here today to talk about gut health, but specifically building it during the infancy stage. So Dr. Mueller, welcome to the show.
Thank you so much for having me.
As we start this conversation about infant gut health, tell us how you actually got interested in the connection between gut health and human health. And I'd like to sort of get into the gut microbiome and all of that, because I don't necessarily think we all understand it as well as you do.
So thanks for the question. So I did a postdoc at Columbia University after a PhD in epidemiology. My background was in nutrition science. So I kind of married the two together during my postdoc and studied the molecular determinants of early life risk for later life obesity. And I emphasized my research on the microbiome. It was a time in which we were learning a lot about the microbiome because of the advent of the sequencing technology that was developed by Rob Knight. And I had the opportunity to collaborate with Marty Blazer and Maria Gloria Dominguez-Bello at NYU. So during my postdoc, I looked at how the bacteria from our mothers colonize the gut and how that could be disrupted by C-section delivery. And then I looked specifically at how that disruption can lead to a differential risk of childhood obesity. So I was previously interested in early life determinants of health because it can facilitate primary prevention of disease. So what factors can we focus on to prevent obesity in the first place? Because we know it's extremely challenging to get people to lose weight once they're overweight or obese later in life. So prevention is the key. And the microbiome really allowed me to look at an early life factor that is seated at the beginning of life and look at ways that we can potentially manipulate that to prevent diseases later in life.
So that's so interesting. So for the listeners out there who might not know what a gut microbiome even means, can you sort of help us understand what you're talking about when you say the gut microbiome?
Yeah, sure. So the gut microbiome, sometimes called the gut microbiota, is essentially the community of microorganisms that live in our gut. We have bacteria, but we also have archaea, fungi, and viruses that live in our gut. A lot of the research is really focused on bacteria because of the sequencing technologies. However, there is now an increase in investigators that are also looking at fungi and the virome in addition to bacteria. So when we talk about the gut in particular, we have a dynamic community of microorganisms that change throughout life. In the first 2 to 3 years, it's a very dynamic population. So it's changing, it's getting colonized from different sources, and it's also getting selectively pressured to increase or decrease in terms of different populations from things like breastfeeding, formula, if you introduce formula, or introduction to solid foods.
Mm-hmm.
In the adult gut, we're talking about 300 to 400 different strains of bacteria that live in our gut microbiota typically. However, that can vary from person to person and also from different stages that you may be in, or if you've taken antibiotics, it may be greatly reduced.
So, if you go on Google and you search gut health, there's all kinds of articles. 7 ways to improve your gut health, 7 things that will hurt your gut health. What is the connection between the gut and your overall human health?
Well, that's a complicated question. And if anybody knew the answer, I think that they would be doing very well in their research. We're still trying to understand the intricacies of the relationship between the gut and health. But as we learn more, we're realizing that it is a very important component of health and helps to determine health from disease states. So there was a recent paper that looked at how well, in a population of about 5,000 individuals, how well all of your bacteria could predict whether you're in a state of health or disease, looking at several chronic diseases, including obesity, type 2 diabetes, hypertension, and other chronic diseases. And that study found that they could narrow it down to about 40 different bacterial strains. And based on the relative abundance or the percent that each individual had of these 40 strains, they could predict with about 75% accuracy whether or not a person had a disease state or a healthy state. Now, that's only, 75% accurate. That means they're getting it wrong about 1/4 of the time. And that's also only a cross-sectional study. So it doesn't tell you whether or not the imbalance in the bacteria preceded the disease state or was caused by the disease state. So while it can be used to facilitate disease prediction, whether or not it's a causal agent or earlier preceding the disease state is still unknown for many diseases. And that's where a lot of the research is focused right now.
Interesting. And so you mentioned obesity and you've mentioned cardio-metabolic diseases. What type of diseases are we talking about in terms of that study in particular, or are we talking just general you're healthy or generally you're not healthy?
So I think specifically with the microbiome, we're typically talking about, so the microbiome is related to immune-mediated diseases, metabolic diseases such as obesity, type 2 diabetes, cardiometabolic diseases. We typically include in that type 2 diabetes, obesity, hypertension, and cardiovascular diseases. In that particular study, they were more interested in cardiometabolic diseases, which are the most common types of diseases in adults.
Okay. So since we're going to talk about infancy, let's get back to, in utero, a baby is developing, the gut is developing. Is it blank? Does it have a microbiome from the mother while it's developing? What is the whole process for sort of getting the infant gut seeded with all that good bacteria that you're talking about?
Jill, that's another great topic. It's actually the topic of a recent grant that we just got funded to look at what are the maternal determinants of the infant gut microbiota. Specifically, we're going to better understand the vertical transmission process from mother to baby, whether the bacteria comes from the mother's gut or vagina, and at what proportion from each of those two different body sites. But we do know already that there is a higher concordance or sharing of bacteria between mother and her own baby than there is between a mom and a different baby, or a baby and another baby, or a mom and another mom, right? And we also know that is, there is a genetic component, but it's largely determined by the environment. So we're interested in to to better understand whether that's coming from the vaginal microbiome or the maternal gut, so that we could potentially in the future learn to optimize that microbiome, the maternal gut or the vaginal microbiome, so that the baby could be seeded with an optimal set of bacteria at the beginning of life or other microorganisms. So I'm going to back up a little bit because the first part of your question talked a little bit about when that seeding process happens.
Yeah.
So in 2014, there was a famous study that came out from Kirsty Agard and colleagues at Baylor Medicine, challenging the notion, the previous notion that the in utero environment is sterile. So that had been a common kind of knowledge among obstetricians and gynecologists that the in utero environment was sterile. So she published some work showing that there was some bacteria or bacterial DNA present in the placenta. And she challenged this dogma, saying that potentially there is a sharing of bacteria between the mother and the fetus before birth, and that we should start to look at what are the sources of the bacteria that might be shared before birth. Since that paper was published in 2014, there have been several studies that have looked more closely at the methods and found that many of the bacterial DNA that were found were likely from contaminant sources that could have come from either the collection and storage of the placenta or from the actual sequencing processing, whereby the kits that were being used or the processing of the samples may have introduced some of the bacterial DNA that was observed.
Gotcha.
So the consensus most recently is that likely those bacteria that were observed were contaminant, and the seeding of the infant gut microbiome happens at birth, right at the time of parturition, and continues, only continues to expand after birth.
Okay. So that's really interesting. So we have two types of birth, right? It's a vaginal delivery or a C-section. What do we know about seeding the gut microbiome between those two very different ways to birth a baby?
Yeah. So one of the major determinants of the early neonatal microbiome is delivery mode. And we've studied this with several different populations, and also looked at other publications and found it to be quite consistent. One of the consistent patterns that we see is vaginally delivered babies are colonized by bacteria known as Bacteroides, which is a common bacteria in the adult gut microbiome, but is also much more prevalent in vaginally delivered babies. So vaginally delivered babies have more Bacteroides, and they have lower proportions of Staphylococcus, which are a bacteria found in the maternal skin. They also have lower proportions of Enterobacteriaceae. And this pattern has been observed not only in our studies, but other studies. A recent study, I think from 2021 in Gastroenterology, the journal Gastroenterology, they looked at a longitudinal cohort of infants and first documented that C-section delivery was associated with greater BMI in the children at age 3. And then they looked at the bacterial populations in the infants at 3 months and 12 months and found that a ratio of Enterobacteriaceae to Bacteroides was on the pathway or on the causal pathway between C-section and obesity, suggesting that this ratio of bacteria was potentially what we call a mediator or something that could be intervened upon. So that work is fascinating and needs to be confirmed with other studies. And we're hoping to look at that ratio as well as other bacteria in a randomized controlled trial that we're doing now to look at whether seeding of a C-section delivered infant can help to ameliorate diseases that are associated with C-section delivery.
So what I hear you saying is that some of the research that is out there is showing that babies born by C-section may have obesity as a result of their gut microbiome. Am I interpreting that right?
You're interpreting that correct. Yeah.
Okay.
So there's been a lot of work that has, including our study in 2015, when I was a postdoc working with Maria Gloria Dominguez Bello. Our work showed that C-section was associated with approximately a 65% increased risk of childhood obesity at age 7 in a longitudinal study. And this is a longitudinal study that was in northern Manhattan and the southern Bronx. And we also found, independent from C-section delivery, that prenatal antibiotics were associated with increased risk of obesity. And the hypothesis was that these two factors are disrupting the mother to newborn exchange or transmission of bacteria and leading to a dysbiosis in the infant, which then subsequently can lead to the development of obesity. There's been several or many other studies, including at least 2 meta-analyses, which is an analysis of all the population studies that had been done at that time, that have shown that C-section is associated with increased risk of obesity or overweight and obesity in children. So now there's an effort to better understand what is the mechanism that's driving this association. Is it a bacteria, or is it other microorganisms that are out of balance, or is it some other factor that we weren't thinking about? Potentially epigenetics because of differences in the stress hormones that are associated with the delivery mode. So there's several studies that are looking at some of those other factors because it's not enough to just show an association or a correlation. We really want to know what is the mechanism that helps us with causal inference.
Yeah. And so is it fair to assume that with a vaginal delivery that the mother is transmitting her colony of bacteria to the infant, and that is a better ratio of healthy bacteria or bacteria that is not associated with the development of childhood obesity?
So that is the hypothesis, but as you can imagine, it gets more complicated the more you think about it, because a mother may, first of all, we don't know exactly whether it's the vaginal microbiome or the mother gut microbiome. There was a recent study that looked at whether seeding infants with the maternal gut microbiome, so giving them a small amount of the maternal stool microbiome mixed with breast milk at the beginning of life. This was C-section delivered babies who were scheduled C-sections, whether that could also alter the microbiome and make the microbiome of those C-section babies more like their vaginally delivered counterparts. This study, I should say, followed a previous study by Maria Gloria Dominguez Bello, which showed that C-section delivered babies that were seeded with vaginal microbes had some restoration that made their microbiome more similar to vaginally delivered babies. And so this investigator, this more recent study, wanted to see whether the fecal microbiota from the mother could also do that and potentially do it more so than the vaginal microbes. And what they did find was that there was, first of all, engraftment of the microbes. Those maternal microbiota were able to colonize the C-section delivered baby's guts and stay around. And it also reduced or helped the kind of convergence between C-section delivered babies and vaginally delivered babies. The convergence happened much more quickly than it would have happened had these two groups not been seeded with the bacteria.
So interesting.
So I think that opened the question for us is like, which is more important, the maternal gut bacteria from the mother or the vaginal bacteria? And at the time of birth, during a natural birth, the vaginal bacteria actually start to look a lot more like the gut compared to other times during the maternal life course. So the change in hormones that happens around the time of delivery may facilitate this convergence of those two different microbiomes at the time of delivery so that the baby is seeded with the bacteria that are needed for health. Now, one other factor that I think could be interesting in the future is what does the maternal health status at the time of delivery have to do with the bacteria that she's sharing? Do obese mothers, do type 2 diabetic mothers, do hypertensive mothers share with their baby bacteria that could propagate that type of disease in the baby in the future? That we don't know. That will be something that will have to be investigated. And along those lines, will there be a certain set of bacteria that would be optimal for the seeding of the vaginally delivered baby?
Wow, it's all so interesting. Honestly, when you think, you start thinking about, well, a lot of us are talking about childhood obesity now, and we're talking about the heritability, the genetic, the multiple genes that are associated with this. And then you talk about lifestyle, and then you're talking about diet, and it's like, and the nutrition component. But to think about the avenue of birth as a powerful influencer on the development, potentially a powerful influencer on the development of childhood obesity, and the ability to actually perhaps change that pretty quickly in real time, right? That's pretty fascinating.
Mm-hmm. I agree.
Yeah.
I completely agree. And when I talk about this, I also don't mean to kind of undermine the importance of diet for childhood obesity or lack of movement or sedentary behavior. That clearly is very important for being a deterministic factor of whether a child's going to be overweight or obese or not. We now have 2 children, a 3-year-old, a 5-month-old. We're just trying to make sure that we're doing the lifestyle habits that we know to be healthy for them, trying to not focus so much on necessarily what their BMI Z-score is or their percentile, although they're doing fine. But I think one thing that gets lost is it's not either or between what is the role of the microbes or what is the role of nutrition and physical activity for obesity. It's actually an interaction of the two. So when we think about the microbiome that's seeded early in life, and then the factors that might disrupt that, such as C-section, antibiotic, or formula use, or something that we haven't talked about, early introduction of solid foods, which can kind of mature the microbiome more quickly. When you're changing that microbiome, the backdrop for metabolism, and then later in life, introducing foods, those same microbes that you're altering early in life are the microbes that are breaking down our foods, changing them into metabolites that are then used throughout the body to alter our different disease states or a risk for disease, or cross the blood-brain barrier and change our mood, or lead to energy. Short-chain fatty acids are a big component of energy for humans. So the same nutrition that's given to 2 babies that have a different set of microbes could lead to very different results. And I think that that's where the interaction between our microbiome and these things that we know to be healthy can play a big role.
And now a word from our sponsor. Making sure you and your family stay healthy means having regular checkups, getting enough sleep, exercising, and eating the right foods. Drinking 3 glasses of milk a day is part of a healthy diet, especially for young children. With 13 vitamins and nutrients, it's hard to beat milk's nutritional value at any age. For more information, visit southwestdairyfarmers.com. So let's segue into formula, because I'm interested in hearing, what's known about breastfeeding versus formula feeding when it comes to that gut microbiome.
Okay, so I think that this is an interesting topic. It's I wouldn't call myself a breast milk microbiome expert, but I'll give you kind of just some of the things that I think are important. So human breast milk is our first kind of superfood that we'll encounter. So an opportunity to be exposed to that as a baby is a good opportunity. And if a mother does have that ability to do that, it's something that's likely very nutritious and natural for the baby to consume. Why is it nutritious? Not only does it contain the macronutrients that we know to be good for health in sufficient quantities for the baby, it also includes human milk oligosaccharides. And it contains over 100 types of these oligosaccharides, which other mammals that have breast milk contain much fewer of. So humans have a much higher concentration and number of human milk oligosaccharides than other mammalian species. And what's special about these HMOs, human milk oligosaccharides, is that they can only be broken down by our bacteria in our guts. They would otherwise not be useful for us as a host for energy or production of other metabolites. So those human milk oligosaccharides are broken down by our bacteria and then converted into metabolites. Some people call these postbiotics. And those postbiotics or metabolites have downstream effects, both on the cells that are in our gut. They also go into circulation, can be bound to receptors ranging from receptors in our liver to also receptors in our nephron, and can also cross the blood-brain barrier and again affect mood. So those are just some of the things that I think are interesting about breast milk. Breast milk also is dynamic, changes throughout the life course of the infant to meet the needs of the infant. What's difficult about formula is that formula needs to be made to try to mimic breast milk as best possible, but that's difficult because it's not precision formula. It's formula that best fits the needs of infants from 0 to 1 year of age. And it doesn't follow that same dynamic process that breast milk does, unfortunately. It's also difficult to create the variety of human milk oligosaccharides in formula to mimic those oligosaccharides that are in the breast milk in formula. But there is an effort to try to do that. I know that there's some companies that at least have one or more oligosaccharides in their formula. And there's a similar type of sugar called a galacto oligosaccharide that can be found in breast milk, in formula, that may be able to come close to having some of the same nutritional benefits as human milk oligosaccharides. So these different types of nutrition have a pretty strong effect on the microbiome, probably the strongest effect in that first couple of months of life after the baby's been delivered. So breast milk has a selective pressure to increase the populations of certain bacteria, in particular, increase the populations of bifidobacteria. And formula can increase the populations of other types of bacteria, ranging from Enterobacteriaceae to Enterococcus, and some of these other types of bacteria that maybe aren't as beneficial or helpful for the baby. That being said, there's still, I think that the evidence base still needs to be developed further. I don't want to, if a woman can't breastfeed, she can still do other things to bring her baby up to speed with a breastfed baby. There's also opportunities.
You mean a formula? Up to speed with a breastfed. Yeah.
A formula-fed baby can nutritionally be up to speed with a breastfed baby. Through following some of the other things that we've already talked about for minimizing risk of obesity, for example, physical activity, a good diet later in life. So formulas come a long way too, and women who can't breastfeed, their kid's gonna be fine, so they don't have to worry either. We're just trying to better understand these different, especially the components of breast milk, so that we can optimize formula for those women to try to mimic the effects of breast milk.
So yeah, and I hear you. That's really important thing for people hearing. The research that you're finding, that you're doing on breast milk, and you're learning about breast milk, informs formula creation or manufacturing to better match breast milk, so that all those benefits can occur to all babies, for all babies.
Exactly. And there's also some new types of probiotics out there that may be beneficial for, depending on what your exposures were in utero and at the beginning of life. So those are worth keeping an eye on. One particular strain I think that you should look for in your probiotic is Bifidobacterium longum infantis, which is one of those bacteria that I talked about that can break down human milk oligosaccharides and convert those into those short chain fatty acids that are beneficial for health.
So this would be a probiotic component for infants.
Yes.
Correct.
So
yeah.
There's a probiotic that's on the market developed by some investigators from University of California, Davis, and I believe it's called eVivo. Mm-hmm. And they've done actually a few randomized controlled trials and shown that the probiotic does reduce inflammation and does lead to improved gut health for infants. In particular, it does really well with preterm infants who are typically C-section delivered and have been exposed to different types of antibiotics. So that's showing a lot of promise. I think there's going to be other strains that you'll see continue to come out in the probiotic market. Just make sure that they're tested. There's been randomized controlled trials, because the regulation in the United States on probiotics isn't as good as it is in other places like in European countries.
Okay. So what I hear you saying is that is yet another avenue. If a family is using formula for their infant, a probiotic might be a good addition to their overall feeding plan, if you will.
Yes, I think that's something that there's a hope for that. The one probiotic that I did just mention, eVivo, that one does best, and I think maybe is recommended to be mixed with breast milk because of the bacteria that's in it. That particular bacterial strain breaks down the human milk oligosaccharides. So it's most beneficial when it's mixed with breast milk. But I think down the line, you'll see probiotics that can be combined with formula as well to get try to mimic the benefits of breast milk.
Yeah. Yeah, that's an interesting area. Yeah, that could be an interesting horizon. What about, you did mention antibiotics, and sort of walk me through sort of the scenario where antibiotics would be used and their impact on the gut microbiome in an infant. I think you mentioned a mom who might need antibiotics during pregnancy or delivery or.
Yeah. So antibiotics in the U.S., the standard of care the prophylactic treatment for a C-section delivery in the United States is to give antibiotics about an hour before the C-section to prevent infection of the surgical wound. So the mothers that are delivering by C-section, elective C-section, are almost always exposed to some level of prophylactic antibiotics. So anything that we're observing with differences between the C-section and vaginally delivered babies is probably a combination of C-section delivery and prophylactic antibiotics. Another use for intrapartum antibiotics, the antibiotics that are right around the time of birth, is group B streptococcus, which is common, a prevalence probably of close to 30% in the US. And women with GBS in the US are prophylactically treated with antibiotics so that they don't vertically transmit GBS to their infant. GBS is a known risk factor for meningitis, and we want to make sure that we're preventing the vertical transmission of that to the infant to prevent meningitis.
Sure.
So that's another scenario where you might see the use of antibiotics. However, there has been some evidence from other countries that potentially using antibiotics after birth for C-section delivery and minimizing use of antibiotics for GBS can have similar outcomes for the mother surgical infections and also for the infant. So I think when we talk about antibiotic stewardship and trying to reduce the number of antibiotics used, those are some approaches that we might learn from other countries about. I think that, reducing antibiotic use is always a good thing, not only for the mother and the infant, but also just for reducing antibiotic resistant bacteria, which are a growing problem, as you probably know. Of course, there's any time that a pediatrician sees your child and recommends antibiotics. I'm not an MD, so follow the recommendation of your pediatrician. If you really feel strongly about not using antibiotics, maybe get a second opinion. But it's never good to start a dose of antibiotics and not finish it. You want to make sure you're following the recommendations. That can just lead to more antibiotic resistant genes.
Yeah. And then you also mentioned early introduction of solid food. And I really want to understand this because, we're sort of focused, public health messaging is focused on introducing solid food at 6 months. And so are you suggesting that early introduction of solid food is a good thing or it's not a good thing on the gut microbiome?
So I'd like to stay away from good, bad.
Sure.
But I think, what
helpful,
we've shown,
unhelpful.
Yeah. So what we've shown, and a lot of this work was done by my PhD student, Maura Differding. She led the work looking at complementary feeding and timing of complementary feeding and how that impacts the composition of the infant gut microbiota, both at the time of introduction to feeding and then also at about 12 months. And so what we found in two different populations, we looked in a biracial population from North Carolina that was lower socioeconomic status. And then we also looked at a population from Quebec, Canada that was higher socioeconomic status. And we found in both scenarios that timing of complementary foods, in particular solid foods, the earlier the introduction led to a disruption of bacteria composition. And looking more closely at kind of the infant microbiome from 0 to 1 year, we found that the early introduction of solid foods led to a rapid maturation of the microbiome. So there was more butyrate producers, which are typically found in adults, and we found them in infants already at 3, 6, and 12 months. One particular species was Lactospiraceae glaudia, was increased in relative abundance. And that also is, again, short chain fatty acid producing bacteria. We also showed that C-section delivery and early introduction of solids increased microbial diversity early in life at 3 months. So normally we think of gut microbiome diversity, higher diversity being good in adult guts. That seems to be the case for many health outcomes. In infants, in early infancy or neonates, in the first 3 months of life, it's starting to show more and more that high diversity of the gut in these infants is not necessarily a good thing. It's been associated with later life obesity. It's been associated with other health outcomes, and it's been associated with formula use compared to breastfed babies, and early introduction of solids and C-section delivery. So those factors, which we know to be kind of disruptors, are associated with higher diversity in the first 3 months of life. Why is that? Typically, typically the human body tries to reduce the number of bacteria in infants or neonates that are still immunocompromised. They're trying to keep it to a select few bacteria that are beneficial bacteria, such as bifidobacteria, and reduce the number to prevent potential infections. At least that's a hypothesis. And when you see a much larger diversity, that may indicate greater immunocompromised status for the baby, or it may indicate that the baby is not getting breast milk or is not getting a heavy set of microbes at vaginal delivery and is therefore more colonized by bacteria that are coming from other environmental sources instead of preponderance of bacteria from breast milk or from vaginal delivery. So it's not exactly clear why this higher diversity seems to be associated with poor gut health in babies, but the evidence is starting to mount to suggest that's true.
And is there a relationship with allergies? I mean, I guess what stood out to me when you were just speaking was this idea of the full immune system isn't fully maturated. I mean, I know from what I understand until age 5 or 6, that's why we say don't feed raw fish. There's like a lot of sort of recommendations around making sure when you're feeding certain foods that could contain contaminants like bacteria that could make a child very, very ill, that, you want to make sure that their immune system is fully maturated. And I guess that's what jumped out at me. I was wondering, is there a connection between just the health of the immune system, allergy development, anything like that you're seeing in this gut research at all in infants?
Yeah. So I want to say two things here to make sure that it's clear. I'm not trying to make recommendations about when, like what age somebody should introduce solid foods. So our study showed that early introduction before 3 months of age or before was associated with differences in the microbiome composition. Whether or not those are good or bad, we can't necessarily say, but there was changes in the microbiome composition that indicated that the early introduction matured the microbiome more fast than if they were introduced to the solid foods later in life, which makes a lot of sense because the microbes that are found with breaking down solid foods are typically found in the adult gut microbiome. We also found that the early introduction of solid foods, this is 3 months or before compared to 4 months or later, was associated with higher BMI at age 5 in this particular study. So I think one of the risk factors for early introduction of solid foods is a higher BMI or higher risk of childhood overweight and obesity. The evidence is still inconclusive, I think, in that area, but there's evidence to suggest that may be true. And that's probably, it depends on the type of food environment that baby is in. If you're introducing early solid foods and you're in an obesogenic household, you're only getting unhealthy foods, then I think it's probably not a good thing because you don't want to be introducing them to, fast food or highly processed refined grains early in life. That's just going to not only change their microbiome, but also provide them with excess calories that would potentially lead to rapid weight gain. Now, on the flip side, I think that there's some pretty interesting research suggesting that introducing babies to certain foods between 5 and 6 months to kind of give them that exposure to the food and potentially allow them to develop some type of recognition of the food so they don't develop allergies later in life. That is important. Not necessarily that they're getting much calories from that food, but to just expose them so that they're not potentially to reduce their risk of having an allergy later in life. And so, the best example of that is peanut exposure. You want to expose your child to peanuts earlier so that they don't develop a peanut allergy later in life. And there's been some evidence from a randomized controlled trial showing that is true. And other allergenic foods as well should be introduced at a certain time point to reduce allergies later in life. But the type of exposure that you're usually getting in that early introduction to solid foods is usually not enough that's going to really impact the microbiome, because
Gotcha.
the child's probably not eating a lot of calories of that food.
Yeah. Yeah. So this has been so interesting, and it just sounds like there's just so much amazing research going on about gut health in infants. And so as we wrap this up, parent to parent, what would be your top tips for a parent who might be expecting a baby, or who might have a young infant in the house? What would be your top 2 or 3 recommendations for those families based on, what you know about all of the evidence that's happening, or your gut instinct on what to advise?
So, well, my advice is to make sure that you're playing with your baby a lot and getting them moving around. First of all, whether or not that's going to help their microbiome or not, which it may, it's good for the baby and keeps them excited. So, doing the things that to be good for your child, of course. Nutritionally, breastfeeding if you can, trying to increase the exclusivity of the breastfeeding, doing your best to do breastfeeding directly from the breast versus expressing and through the bottle. There's some evidence that there may be differences in the types of microbiota that are shared with the baby if it's directly from the breast versus from the bottle. Mm-hmm. Whether or not that has any impact on the child microbiome is still unknown, and also whether or not it has any difference for child risk of disease is unknown. But I think that there's kind of just a natural connection between the baby and the mom when that happens. Reducing use of antibiotics if possible. Again, this is going to be dependent on the types of exposure that your child might have. Many of those exposures come if they're with a nanny and other kids, or if they're in daycare and out of your control. So do the best you can. Don't be too hard on yourself. Mm-hmm. You may also consider doing some sequencing or examination of your child's microbiome at some point in the first 6 months of life, and seeing what the balance of bacteria is, especially if you've had a C-section delivery, if you have been only able to do formula feeding, or your child was exposed to early antibiotics, and you want to see whether or not that disrupted the bacteria enough so that you might consider using a probiotic. One of these probiotics that I suggested on the podcast could be beneficial to increase the numbers of bifidobacterium, mm-hmm. which could be beneficial for the child. So maybe a screening could be helpful. They're coming very inexpensive. You can even participate in many research studies if that's something that interests you. It's not going to be paid for by insurance yet. It may in the future, but it's something that will give you some kind of insight into what's going on in your baby's gut.
Yeah.
So I've given you a couple of things. I also think just being cognizant of making sure your baby gets outside and is playing a little bit. They say, eat dirt. There's a book about eating dirt. I wouldn't necessarily say eat dirt, but don't be the type of parent that doesn't allow your baby to be exposed to some dirt every once in a while, and don't overuse the cleaning agents in your house that are antibacterial, so that your baby's exposed to some natural bacteria that way as well.
So
I'll leave you kind of with an example of a very high microbiome diversity population, and that is the Yanomami Indians down in Venezuela, on the Venezuelan-Brazilian border. I had the opportunity to work with Maria Gloria Dominguez Bello, and she has done a lot of research with this group of Indians, and found that they have the highest microbiome diversity in the world. They also have the lowest blood pressure in the world, which is a finding that I published on in 2018. And there's some evidence to suggest that there might be a link between the microbiome and blood pressure. But what I find fascinating is that they have the highest microbiome diversity, yet they eat a very low kind of range of foods. So the diversity of their foods isn't that great, but they do play a lot in the dirt. They eat a lot of resistant starch throughout their life, a lot of natural prebiotics and resistant starches in the forms of, plantains and bananas and other things.
Mm-hmm.
And so what exactly it is that's giving them that high diversity, it's still not known, but I would consider exposure to a broad range of environmental exposures and eating a highly fibrous diet when your child can have fiber foods, one that's high in resistant starch in particular, that can help to feed the microbiota and hopefully give you good gut health for your baby.
Really fascinating stuff, Dr. Mueller. Thank you so much for coming on the show and sharing your expertise with us. If folks want to get in touch with you or follow up on maybe enrolling in a study or something, how can they find you?
So they can feel free to give me an email. I can send you my email and you can put it in the show notes.
Okay.
I also have a website. You can look me up through Johns Hopkins Bloomberg School of Public Health, where I'm an assistant professor. And I think one of those ways should be fine. And I would love to follow up with anybody who watches your show.
Awesome. Thank you so much. This is like mind blowing. A lot of this research. It's such an honor to have somebody who's so well versed in the research to come on the show and really come at it from a very authoritative angle. So I really appreciate that.
Well, thank you so much for having me on, Jill. I'm glad that you're having people talk about these important topics. And I hope everybody enjoys the podcast.
Thank you.
Thanks for listening to the Nourished Child Podcast, where the #1 goal is to help you grow a nourished child inside and out.